Okay, let's start the second little lecture to follow up on what I said I was going to do, which was to talk about cell specialization. We left by looking at this anatomy site uh, that you can use for certain things to study anatomy and even function. But in our case, there's a link to cells, general structure that you can look at there. We're going to look at neurons and nervous tissue as really an individual type of cell that or tissue that contains several types of cells, and they're all specialized, so we can, get an idea of what's going on there. First of all, I have a bunch of resources here that I can use to help you look at these cells. Okay, first of all, let's look at this particular cell. It's called an astrocyte. This is a diagram of a tissue that is called glia or neuroglia. That's like looking at the smashed soft tissue of your brain. And most of the tissue of your brain is there to support the functional cells, which are called neurons, and we'll be looking at those in a minute, but this of course points to this neuron. There's the nucleus of the neuron. This is an astrocyte. What's significant about it is, if you'll notice what it's doing, it is holding blood vessels and neurons apart. If you think of when you look under the hood of your car, there's usually a bracket that holds the different spark plug wires apart so that they don't touch each other, rub on each other, and short out. You have the same thing happening in the nervous system in your body, in your central nervous system, in your brain. And the interesting thing about this astrocyte is, we can look at some organelles there, mitochondria for energy, the nucleus is there. What they are not showing that is particularly interesting to me about this cell is the cytoskeleton. So when you learn about the cell, you'll notice when you look at different animations or when you read your book, there's a cytoskeleton. There's proteinaceous fibers inside of the cell that give it structure. Well, that doesn't mean too much in a generalized cell until you understand that the structure of some cells is very important. In this case, this astrocyte has to have a rigid structure to hold blood vessels apart from neurons and neurons apart from other neurons. Otherwise, like in the walls of your home, if hot wires short out, a fire can start. At very least, your breaker will break and you won't have that function until you repair the damage. So that's one of the important things to think about, is that these cells, these astrocytes, which are a part of the neuroglia, are there to provide really a structural insulation of nerve cells from each other so that the action potentials, or electrical impulses, if you will, can safely go down the axon and the message will get to the right part of the body. So let's look at another diagram, and that is one of an individual axon or neuron. Actually a neuron, a neural cell. Okay, the neural cell has all of these parts, the nucleus, cell body is here, and they have some unique features, but on this end there are dendrites. So the message that comes from the other neural cell comes in to this part of these dendrites. Let's say that let's say that these are coming from touch receptors. Well, you might get something that touches you in the air, and it only touches that one little dendrite. You'll never feel it. You'll never know it's there. But what if the touch is hard enough, like if I poke you with my finger, to stimulate a lot of dendrites at one time? Then all of those impulses go into the cell body, and that results in an action potential or an electrical impulse being large enough to be propagated and sent down the axon of the nerve cell. This axon might originate at your spinal cord around your hip or above your hip and go all the way down to your toes. So this part of this single cell can be 3 feet long or longer. And it's going to go down and branch and be there to stimulate muscles, probably in this case. You'll also notice that this axon is surrounded by Schwann cells. Schwann cells are a unique cell that has a differentiated membrane. Not the same as the general membrane you study when you study the individual general cell, but a membrane that is myelinated. It is a very fatty, tough substance that wraps around the axon. And by the way, these little purple things are the nuclei of the Schwann cells. And they wrap around in such a way as to provide an external fatty myelin sheath to protect the axon from shorting out. So it's like taking black tape, black electrical tape, and wrapping it around a bare wire to provide insulation. And this insulation occurs around the cells in your brain, in which case they are called oligodendrocytes. And if this is outside of the central nervous system, then the cells are called Schwann cells, and they're a little different. And the reason they're a little different is instead of being continuous like oligodendrites are, there are these nodes of Ranvier. Cool. And these nodes of Ranvier are unmyelinated. And what this structure allows for is for the action potential to jump from node to node, which is faster than running continuously down the neuron axon. So it provides for a quicker transmission of information when the distance from the central nervous system is longest, which it is outside of the spinal cord or brain. The distance is longest, so by saltatory action, jumping from node to node, the impulse gets to the muscle or the gland more quickly. And in case of a gland, there's even a different kind of axon terminal associated with the cells of the glands, so that instead of a chemical transmitter from nerve to cell, there is an electrical transmission from nerve to cell, so that those actions take place faster. And those are actions like secreting insulin into your bloodstream. And the insulin from the pancreas, from specific cells in the pancreas, is secreted. How do you think? Well, in general cells, you will learn about exocytosis, which is a way that several things happen here. One of the things that happens is there is a neurotransmitter, generally, that diffuses between the axon terminal and, say, the dendrite of the next nerve. That neurotransmitter diffuses between those two nerves by diffusion but it has to get out into this little space between them first, and that happens through exocytosis. There are vesicles containing neurotransmitters that actually move to the cell wall and spit out those chemicals. And then there's another place where this is very important, and this is in the cells that are differentiated to be glandular cells that secrete hormones into your blood, like in the pancreas that I just talked about. In that case, all of these cells simultaneously move vesicles to their cell walls and spit out insulin so that enough of it gets in your blood at one time to give the desired effect, which is to get sugar into your cells, because that's their source of energy. That's the food for the cells. So you can see there's several types of differentiation here. There's differentiation of vesicles and exocytosis in two different functions that I just talked about. There's differentiation of cytoskeleton in the body of the astrocytes that I talked about in the previous slide. And then there is also some differentiation of cytoskeleton in the nerve cell to maintain the branching spread of the dendrites, which bring sensation into the body of the cell, or information in, regardless of what kind of sensation that is. And so this type of cell would be both outside of the central nervous system, say on your skin or in your eyes, your nose. Think of your senses. It would also be within the nervous system where other types of communications happen. So that's probably all we need to talk about, but I wanted you to appreciate the fact that from the general cellular structures that you learn and cell organelles that you learn about what they do, you'll find that there are specifics. I'll give you one other example. Mitochondria are the cell bodies or the cellular organelles inside of each cell that provide the energy that make ATP, adenosine triphosphate. Well, our mitochondria, every one of them in our bodies, came from our mothers. And so people are genetically looking at mitochondrial DNA as a way to trace our lineage back all the way to Eve, in theory. And you would ask then, why don't we have mitochondria from our fathers? We'll learn about that in, I think, Unit 6. But the reason is that mitochondria in sperm are in the tail. They're used to produce all this energy that's needed to swim towards the egg. And once the sperm fuses with the egg, the tail falls off. So most of the time, we only have mitochondria from our mothers. And so there's a specialization there where these mitochondria are very abundant in the tail of sperm cells. Lots of variation occurs in the tissues in our bodies that we don't appreciate as we study only the general structure and anatomy of a cell. But I wanted you to think about that and hope that you'll get a greater interest in the cell structures and organelles by listening to this lecture. And then perhaps when you look at these structures and read about them, it'll be more meaningful to you. Okay, have a good week, have a good unit. We'll see you next week.